Hello. All right, we're going to start with Chapter 1. This is Microbiology for Healthcare Professionals. This is the first lecture. You'll note that in the lecture notes it states that Chapter 2, it's on the student's own time, and that's because basically it's pretty much a biochemistry review that you may have experienced in A&P1. If you find that you're somewhat weak in that area, then I do encourage you to review that because it's going to be essential to understand issues of certain biochemistry, pharmacology, and the structure of certain microorganisms. So let's get moving into this because we've got a lot to go through. Now you'll notice also in the lecture notes that it's presumed that you've taken time to review the key terms as well as review the glimpse of history. Those are going to be very important to introducing you to the concepts for each chapter, so I encourage you to do so. Also, you will find from time to time that I'm going to use images, pictures, diagrams, etc., that are not in the textbook. And I will also bring in certain topics that are not in the textbook. So when you're listening to this video, you're encouraged to review or have along with you the lecture notes, because I'm going to be expanding upon certain points, adding certain points to help you to understand the critical importance of certain areas in microbiology. Okay, so let's get moving. First off, what we're going to be talking about is where did we get into this concept of the origin of microorganisms? There was a theory going into the 14th, 15th, 16th century, maybe a little bit earlier, called the theory of spontaneous generation. Now it states that living organisms spontaneously developed from non-living matter. Now we could all sit there and laugh at it, saying, you mean that my brother came from a chair, or I could get puppy dogs from a tree, or even better yet, since a tree is living matter, let's just say, well, you know, how about it from a rock? No, that doesn't work like that, and we'd laugh at it now, but people did believe in this. Particularly, you would see concepts such as maggots would spring out from rotten meat, that eels would come from the lake mud, or excuse me, the river mud. They didn't understand where things existed and in their ecosystems, etc. So basically, they did believe the potential for organisms to spontaneously, just on their own, develop from non-living matter. Francesco Redi in 1663 refuted this theory, and this is his experiment you can see here, where basically he took two containers, one was open, one was covered with kind of a gauze, almost like a cheesecloth, okay? And what he did was show that it was not that maggots spontaneously generated from rotten meat. What happened is the maggots were isolated from appearing, because what happened was the flies that laid the eggs, that the eggs turned into maggots, were isolated from the rotten meat. And interestingly enough, I believe in some of the descriptions of this experiment, the gauze-covered jar, you'd have the flies lay their eggs on top of the gauze, but there would be no maggots in that rotting meat. Okay? Or basically just a regular piece of meat that was just hanging outside. And the seal jar did the same thing. And what he's basically showing is that, look, these things don't just spontaneously occur on their own. The problem came up a little bit later. So therefore, the theory hung around for another 200 years, and we're going to get into Pasteur's work that finally put a stake into the heart of it. But in the process, what helped to keep it somewhat alive, at the same time open up an entire idea of, wow, you're kidding me. There's stuff we can't see, but they exist. And that was Antoine van Leeuwenhoek's work in 1676. And you can see this is one image of it and how it was very simple. It was a very simple microscope. And he was able to see protozoa and small organisms. He called little animalcules. Here's another view that you can see in your textbook. Notice it only has one lens. When you start working with issues involving compound microscopes, keep in mind, compound means more than one. So, compound microscopes in a lot of cases have 2 or 3, may also be assisted by not merely lenses, but also prisms to redirect the image and redirect the light to the eye in a certain format, depending on how it's designed. But I go ahead of myself. Here you can see that Van Leeuwenhoek invented the microscope, saw bacteria. He wrote this. He called them little animalcules. This is actually an image that was reproduced of some of the things that he did. He looked at things from, his own semen. He actually looked at some of the spermazoa there. He actually looked at some of the plaque from his own teeth. He looked at pond water and all these other things. And this was revolutionary because of the fact they'd never been able to see them before. Okay? Now, get to Pasteur's work. Now, Pasteur was still dealing with spontaneous generation. And his work was going to lead to eventually the biogenic theory. He was able to completely refute this because after 1663, when Van Leeuwenhoek, some people went, well, then dead material can still have living organisms. They're just small. We don't see them. So here's what he did. He took broth. Now, broth, and you can make this, we're not just talking beef broth, but you can take like hay, boil it, and it becomes the perfect culture medium for microorganisms. What he did, though, was unique. He took the broth, he sterilized it. He also had the glass shaped in such a way so that basically it was twisted at the neck, if you can see that in the diagram. And so what happened was the bacteria and dust from the air would settle into the bend, but the broth, being allowed to cool slowly, it stayed sterile. There was no organisms. When organisms were eventually cultivated, and I'll tell you that in a second, the broth would get cloudy looking. Okay? Now, they actually have at the Pasteur Institute some of the very same glassware. He was doing these experiments in the 1860s and 1870s, and they're still sterile. Yes, well over 100-something years later. Now, mind you, one of the ways to prove the experiment from the other perspective was the flask was tilted so that the sterile broth came in contact with the bacteria dust from the air, and it brought it back in, and hours or days later, the bacteria multiplied in the broth. The broth was basically very, very cloudy. Another way you could have done it is basically just snap off the neck, and so you had an easy way for microbes, bacteria, etc., to fall into the open container of the flask. So, this led to the development of the biogenic theory. That is, living organisms come from living organisms. And then we get into Tyndale's and Cohen's work. In 1876, they found that different nutrient broths required varied boiling times. This came up with the concept of endospores. Get to know that term, endospores, very, very well, because you're going to find that that's a key point in many microorganisms and how they survive and how they have become pathogenic. Okay? Endospores are a heat-resistant form of bacteria that can survive long periods before germinating. And usually what triggers them to germinate is when the environment's more favorable and they become actively growing bacteria. You're going to see this in Clostridium bacteria, you can find this in Bacillus bacteria, etc. You go ahead a year later, Robert Koch determined that anthrax was caused by Bacillus anthracis. And this was transmitted by endospores of that particular bacteria. Now, if you take a look here, but before we go that, we have to ask the question, the origin of bacteria. At this point, we can have theories and suppositions. Nobody was around 3.8 billion years ago to sit there and say, how did life develop on Earth? But we have, based on various pieces of information, data, chemistry, fossils, etc., some ideas. And the evidence suggests that bacteria developed over the time of Earth's history when the atmosphere was devoid of oxygen, so it was anaerobic, and the surface temperatures, due to volcanic activity, were very hot. And this has probably led to the development of archaea bacteria, which are really not bacteria, but they're called archaea. Archaic organisms, they are also known as extremophiles, and we'll get more into that later. And then, of course, as the Earth continued to cool, you had development of other organisms, and, of course, life goes on. But if you take a look here on this chart, you'll see a lot of the advances in microbiology over the centuries. And you can see how these developments influence things and how fast they would change things. And this is, whether you're talking about society in the sense of population growth, medicine, how we dealt with treating diseases, how we dealt with controlling diseases, how we were able to actually conquer one of the most dreaded diseases, the scourge of mankind, that was called smallpox. And what I encourage you to do is you don't have to memorize every little detail there for like it's going to be on an absolute exam, but you will see a lot of these mentioned later on in various chapters. So yeah, you want to get familiarized with them. Now, how was this progress due? This was part of the scientific method. There are steps that consist of making an observation about something, asking a question about the observation, and then developing an explanation and devising an experiment that will test the observation. So your hypothesis is not an educated guess, as some people would give. It is a testable explanation of an observation. In other words, you've got to be able to test it. Doing the experiment, you collect the data and you draw a conclusion. Communicating the method's results and conclusions. Now, by the way, your conclusion could completely destroy your hypothesis, so you have the opportunity to develop new hypotheses, test them again. So where do we go from here? Well, that's where we get into the term theory. It's an explanation supported by a large amount of experimental evidence collected to support a hypothesis. But notice I put in my notes, but beware. A theory never proves, but only supports an observation. Theories can be disproved by subsequent observations, experiments, or better tools of observation. By the way, you'll notice from time to time that it says microassessment. I encourage you all to, as you read, go back and review each and every microassessment to make sure that you understand them and can adequately answer it. Let's move forward. The activities of microorganisms. Well, much of the aquatic and some land-based production is microbial in origin. And it requires, or I shouldn't say it doesn't require it, it basically creates the conditions that we enjoy in life today, such as oxygen production. Plants perform some of the function, especially in land, but you need to be aware of the fact that some microbes, particularly those in the ocean and freshwater bodies, do also generate oxygen. We also have the recyclement of nutrients. All of this affects our lives. What do I mean by that? Well, think about it. Bacteria and fungi break down all the organic matter. Otherwise, we'd be buried in our own organic matter. Leaves, cut hair, broken branches, and that's just some of the simple things, not to mention, shredded paper and all this other stuff. But then you have also the recycling of the nutrients carbon, nitrogen, phosphorus, sulfur into the environment so that you have this recyclement. Nothing becomes sterile. All of that will be available. Okay? Biotechnology has been benefited by microbiology, using biological organisms to produce materials and services for industry and consumer use. Hey, look at, if you went out and you had your beer or had a wine, or even enjoyed bread, that's all because of yeast. Bacteria can lead to the, different types I should say, lead to the contributions of the creation of sauerkraut, yogurt, cheese, buttermilk. Furthermore, there is a concept called bioremediation, and this is actually using biological organisms, like microbes, certain types, to break down pollutants. Yes, even to break down oil slicks, and to clean up hazardous materials. This has been something that's been done greatly. When the breakdown of the Soviet Union occurred, a lot of former Soviet bases in Eastern Europe were loaded with all sorts of toxic materials. They had spilled jet fuel. They had old broken down materials that were also explosives and things. And they brought in Western scientists using techniques like bioremediation to help clean these sites up. Chemical synthesis. Well, look at this process is biotechnology too. It's used in Japan, it's used in other countries where they make all sorts of different flavors and, ascorbic acid and citric acid. Think about even, the sour candy that so many kids go crazy over. Usually what it is that, yes, there's that sweetness from the sugar, but it's the malic acid created by microbes, the citric acid created by microbes that help contribute to the unique sweet, sour taste of the candy. But we go into other things besides just candy. We're going to talk about this later on, but you're going to find out that Certain microorganisms can produce ethanol, acetone, butanol, antibiotics, amino acids, cellulose. There's a particular type called Bacillus thuringiensis, which is used as an insecticide. Biodegradable plastics. As a matter of fact, there's something biodegradable plastics, polylactic acid, that's used in the 3D printers now. Okay? Genetic engineering. Microbiology has contributed tremendously to genetic engineering. Microorganisms have had their genes spliced or added into their genome to produce various drugs, vaccines, flavorings, enzymes, tissue growth factors, etc. If you take a look at medical microbiology, we've got a lot here because it's crucial for the survival of society, for global society. We transport and move people today, and you can literally go around the world in 24 hours by airplane, and you can go from one place to the other, and this creates unique situations when we deal with immigration, emigration, trade issues, because what also hijacks on those travelers are pathogens, diseases. They can lead to new disease outbreaks, etc. So when we talk about medical microbiology and epidemiology, we're monitoring old diseases, monitoring emerging diseases. EID is short for emerging infectious diseases. You can see that somewhat on the figure there, where there have been different outbreaks, and as you know, they've caused certain situations for the society. Challenges for the global society. Do we allow air travel in? Do we allow air travel out? How do we protect diseases from travelers going to the rest of the population? And we're going to be talking about host-pathogen interactions, and we're going to be talking about how we used to, interestingly enough, think certain diseases were caused by some other factor, but we now find them to be caused by pathogens. One case, Helicobacter pylori, which has been found to be one of the major causes for stomach ulcers. And everybody used to think for decades, and they would poo-poo scientists until some very brilliant scientists down in Australia. Everyone else was poo-pooing, no, it's stress. Stress causes stomach ulcers. And these two scientists went and developed a culture of Helicobacter pylori, and one of them actually experimented on himself. He drank it, and then asked his colleague, I want you to do an endoscopic examination of my stomach afterwards. And it showed the stomach irritation. Because what Helicobacter pylori does is it creates the conditions for the removal of some of the stomach mucus, the protective mucus, and leads to the destruction of the stomach lining. Okay? So, by the way, you'll notice from time to time that I don't go wild with case presentations. That's not that I'm ignoring them. You will have those as assignments. But I encourage you, as you read through the book, to read all of the things, whether it's micro-assessments or case presentations or topic reviews, etc. Now, we've got to start talking about the microbial world. And we have to go into different sort of groupings. And it gets challenging. They used to have the 5 kingdoms, bacteria, and most of the others were the ones with that were eukaryotic. Now, what do I mean by eukaryotic and prokaryotic? Okay, prokaryote are cells without a nucleus. The only groups that exist there are bacteria and archaea. Eukaryotic are cells with a nucleus. Okay, so that includes the other 4 kingdoms: fungi, protozoa, plants, and animals. But what happened was, Wolsey was a brilliant scientist in the 1970s, and he said, look, let's make this into a little bit more easy system. And he used, of all things, studies of ribosomes that were seen to be present in all these different organisms that had cells, and he was able to break them down into domains. And so the major living organisms are bacteria, archaea, and eukarya. Bacteria have single-cell, or basically single-cell prokaryotes. They can have specific shapes: rod, spherical, spiral. We'll get into that in Chapter 3. Most have rigid cell walls. They multiply by binary fission, and many can move by appendages such as flagella and cilia. Archaea has characteristics that are very similar to bacteria: single-cell prokaryotes. Most have specific shapes: rod, spherical, spiral. Most have rigid cell walls. They multiply by binary fission. Many can move by appendages. So it sounds like they're pretty closely related, but when you look at the biochemistry of them, no. Chemical composition of the cell walls is different. In archaea, there's no peptidoglycan, as well as differences in cell membrane composition. Also, their living environments are very different from regular bacteria. They reside in extreme environments, very hot, very cold, chemical extremes, very acidic, very salty, very sulfurous. Okay? We're going to get into that later on. When we talk about eukarya, and you notice in this table here, they're made up of 4 kingdoms. Algae, protozoa, plants, animals. Now, plants we're not going to be dealing as much with. This is not a botany course, but we are going to mention plants in certain situations, and it's important because sometimes some bacteria will act plant-like in the sense that they have certain capabilities to get energy from light, but we'll talk about that later. The unique characteristics that separate eukarya from archaea and bacteria is they all have a nucleus. They all have membrane-bound organelles. Some are single-celled, such as protozoa. Others can be multicellular, such as the helminths. Helminths are those parasitic worms that you might hear about. We'll get more into that later. Algae contain chlorophyll, green pigment, to perform photosynthesis. They have rigid cell walls of cellulose. They live in water. Fungi are a diverse group of organisms. They gain energy from organic molecules, not sunlight. Most live on land. Helminths are a group that contains roundworms, flatworms, tapeworms. Many of them are parasitic. Protozoa are single-celled. They don't have a rigid cell wall. They require organic compounds for energy. Many are motile, meaning that they will move around. They are basically classified based on locomotion, which is how they transport themselves. Both algae and protozoan are also called protists. Now, you can see the 3 domains here in this diagram, and you see the characteristics. And you also notice the typical cell sizes. Okay? Bacteria and archaea are much, much smaller, and eukarya range from 5 to 50 micrometers, and that's what the symbol means there. But you're going to see that there's no peptidoglycan. They can be both single-celled, unicellular, or multicelled. And there's a lot of differences in the eukarya group. We'll go through this as time moves on. We have to be familiarized also with binomial nomenclature. You're going to see this repeatedly. Lots of organisms have a 2-name system of classification. Okay? Binomial, 2. The first name is the name of the genus, and it's capitalized. It can be abbreviated as a capitalized first letter. The second name is the species name, and it's not capitalized. It cannot be abbreviated. The names are derived from the place of the organism was discovered or the name of the discoverer. Note that some species have slight differences genetically and thus are identified by a strain, usually designated by a series of letters and or numbers afterwards. So, Escherichia coli, E. coli, for example, strain K12. I've worked with that strain. Very simple strain. But you might have heard of E. coli O1, if I remember it right now, let's see, 187 colon H2. That's the nasty one that causes death. It's a food poisoning type. But we'll get into that later on. Oh, yes. Let's go back here. Viruses, viroids, and prions. Ooh, that's going to get interesting. The big debate is, are they living? Viruses, viroids, prions must infect the host cell to reproduce. They cannot live and reproduce on their own. They are not made by cells. Thus, these are referred to as obligate intracellular parasites or acellular infectious agents. They must parasitize a cell to hijack the cell's mechanisms to reproduce and function. What about the virus? Well, it's usually composed of a protein coat and a nucleic acid materials inside. That's the genetic blueprint for the function and, in essence, the reproduction also of that virus. It's usually either DNA or RNA. Viruses exist that infect all 3 domains of life. What about viroids? These, interestingly enough, are little loops of bacteria. And I'm jumping ahead here. I should introduce you to these. And that's what I'll do if you'll just give me a second here. If you notice here, this was the binomial nomenclature. You'll see this on table 1.1. Okay? And I encourage you to review that. This is the size variations that you're going to see. And once you get into unaided by the human eye and you go beyond that, that's where you can see why Van Leeuwenhoek had such a great revolution when he was showing his new microscope, because they could see things that couldn't be seen with the unaided, with basically the human eye. Here we have bacteria, the different shapes. You can see rod, round or cock's eye, and then the spirochete or the corkscrew shape. This is algae. This is fungi. There's two different types here. One you see is in sort of a yeast, single cell shape. The other is more in a hyphae structure, a very complex structure with mycelium, etc., and that you would see with certain types of bread molds, etc. Here we have a type of protozoan, and you'd see the little cilia on it that help propel it. Now we're up to viruses, viroids, and prions. Here is an example, and you see it in your textbook, of, I believe it's an influenza virus. Now, that's interesting. You notice that there's that outer coat, and it's made of proteins. So what happens in the DNA and the RNA is they have to provide that information of the blueprint of making those proteins. There's not a whole heck of proteins. It's interesting because the bigger the genetic piece of code for the virus, the less it's dependent on proteins for viral replication or making proteins or other things inside of the host cell. The smaller the viral DNA or RNA code, that loop or single strands, the smaller it is, the more it's dependent upon the cellular machinery of the cell, the host cell that it infects, for its capability to reproduce. I hope that came out clearly. Here you have viroids. They consist of small loops of naked RNA that infect plant cells. Now, there's some scientists that are still speculating the possibility that viroids may cause human diseases. So we introduced this, and you can see basically the PSTV, and basically that's the potato spindle tuber virus. Those are little loops against that big, big loop of T7 DNA. T7 is a bacterial virus. Okay? So you have virus and viroids. The viroids are really teeny weeny, but you get them in there and they travel throughout the stem system of the vascular system of the plant, and they will then cause damage. Okay? The big one that's causing a lot of concern are prions. They consist of misshaped proteins with no nucleic acid found. Stanley Prusiner first discovered this and characterized it in 1982, and 1997 received the Nobel Prize. They act to infect neurons and misshape other similar proteins. They cause neurodegenerative diseases, yes, that's a mouthful, in humans and other animals. For example, you may have heard of Cruru, Jakob Creutzfeldt disease, mad cow, scrapie, bovine spongiform encephalopathy. These are all caused by the prions, and it takes them longer to move through and begin to convert other proteins into the scrapie format, into the prion format. And as this continues, nerve cells begin to die, and so when they take a slice of the brain tissue, they'll notice it's spongy, filled with blank areas. Okay? Now, at this point, you have your microassessment, and then you're to review the microbiome. Here's another image of those prions, by the way. And size considerations. There is a point I wanted to bring that up. You saw that earlier in that earlier image. What you have to keep in mind is this. Most microorganisms are below the range of normal eyesight. That's why, in part, also, Van Leeuwenhoek's invention has become critical for us to understand biology and microbiology. We need magnification tools. The size of bacteria is going to be usually between 10 microns, and remember a micron is the same as saying a micrometer, that's a millionth of a meter, to about 100 nanometers. Nano means billionth, so a nanometer is a billionth of a meter. Human cells range from 0.5 millimeters to about 100 microns. Okay? So what we're talking about is it's going down. Now there are exceptions to the rule that do exist, but the key point is that as you get smaller and smaller, you have to have more sophisticated tools to enhance the viewing of these organisms, let alone study them. Okay? So, I encourage you to review the perspective and the chapter summary. Again, as I said before, Chapter 2, you're on your own, only because we've got so much neat stuff to go through. I would encourage you to move over to Chapter 3, review the key terms and the glimpse of history, and let's start talking about the microscope and cell structure. First off, if you go to 3.1, you're going to see this for review. You got to keep in mind that bacteria are going to be classified according to the following criteria. Much of this we'll examine in this course and in the coming weeks, as well as in the labs. First, morphology, shape, and grouping. Okay? And what I mean by grouping is, are they single cells, or do they kind of bunch together? Staphylococcus aureus. Staph A. Staphylococcus means they're small, round, but Staphylo in this case is they look like a cluster of grapes. So in other words, the cocci, the single cell bacteria, are not solely alone. They're bunched together. They interact with each other and they may be attached to each other. Aureus means gold, and that is because when you look at them under culture, they're going to take a yellowish or goldish appearance. Type of nutrition. This is another key point. You're going to see this coming again and again and again. In biochemical tests, you're going to see this when we talk about in the upcoming chapters. What are their nutritional requirements? Do they get their nutritional requirements for making carbon-based compounds like proteins, fats, carbohydrates, etc.? Do they get it from other sources? Do they make it themselves? Where do they get their energy? The type of cellular respiration. Can they survive in oxygen atmospheres? Can they not survive? That's aerobic versus anaerobic. And then the Gram staining reaction. And also other staining techniques. We're going to see these. These are all going to be helpful indicators for you to isolate and identify the particular microbe. But we are in the age of genetic analysis. And if the organism's genome has been mapped and sequenced, that is, if the genetic blueprint for this organism has been read and cataloged, we're going to probably be able to identify it and identify if it has any strange or unique characteristics it may have picked up, such as antibiotic resistance. Okay? What we're going to go through is the light microscopy techniques, and you're going to see some of those images under the various types. The first type, of course, is basic bright field. What you have is that the basic light will evenly shine through the organism. Modern bright field microscopes consist of an ocular, an eyepiece, and an objective lens, and this is referred to as a compound microscope. The condenser lens, which is located next to the light source beneath the microscope stage, is used to focus the illumination on the specimen. If you look right here, you will see where the specimen stage is. Light is coming up through, and where the specimen stage is where the slide would be. All right? Now, that's pretty cool. We have to get used to certain terms, and I encourage you to get used to these terms quickly. Resolving power is the minimum distance existing between two objects when those objects can be still viewed as separate entities. In other words, this gives you an indication of how clear you can see the images. As you get smaller and smaller, it gets to be a greater challenge. The maximum resolving power of the best bright field microscopes is at about 0.2 microns. You can enhance this, and usually, when you're talking about bacteria, viewing them, you have to go through what is called oil immersion. Oil is used when you have higher powers, 700x and above, since the air between the specimen and the objective lenses would bend the light rays. This is called refraction, and I believe I have an image right here. Take a look there. Now, the upper image shows you if you put a stick, a pencil, or anything else into a glass of water, what happens? It appears as if it bends. That's because light will go through at different rates and different speeds, or will be diffracted when you go from water, glass, and then through air. With oil, you basically keep the light going at about the same speed as it goes through the glass of the slide, through the organism, through the oil, and through the objective lens. And so you have less distortion. And you have to have that. You've got to minimize the distortion that occurs. So that's why oil is used. You, of course, have to clean the oil off your objective every time that you're finished with it. But I'll tell you that using this process, you can see bacteria fairly clearly. You can see them distinctively at magnifications of 1,000 or even up to 16,000 x, okay? Contrast is the number of visible shades in a specimen. Now, since specimen may have components in its cell that have different densities and refractive indexes, it's possible to obtain further contrast of the cell. The way to think about it is this way. If you've ever looked at really, really old glass, like a glass window, you'll see that some of it almost has somewhat of distortion into it. And the problem was before 1850s, it was hard to see if there was anything inside those cells because it was like transparent upon transparent upon transparent. Then we had the evolution of dyes. Most of them were coal-based dyes. This was brought up through some of the German scientists in the 1860s, 1870s. And they helped to enhance contrast because the dyes would attach to certain structures only. The nucleus, the cell wall, the cell membrane. They might attach to endoplasmic reticulum or the Golgi bodies. Or, if you were bacteria, just to the nucleoid or to the cell walls, etc. Selective dyes can stain the structures and create new levels of contrast. Other light microscopes will increase the contrast based on a variety of strategies. If you take a look down lower, you've got dark field. Okay? Dark field is directed toward the specimen at an angle so that the light scattered by the specimen enters the objective lens. You also have phase contrast. Phase contrast amplifies the differences in the refractive index, and you can see right down there. So what you have are these differences enhanced by the specimen, and the denser the material, the darker the appearance of the image. Now they also have what is called interference, differential inference, which is up in the top there. You can also see it much better in your textbook. And this is also known as Normansky differential interference contrast microscope, NDICM, or DIC, I should say. They also call it DIC. You have two light beams. They pass through the specimen and they recombine. Those light beams that are out of phase help to yield a three-dimensional appearance. It's almost like you can see the amoeba up at the top there. It's almost like it's coming out of the actual image. Okay? Moving on. Fluorescent microscopes. Now the neat thing here is you have a fluorescent molecule. It absorbs light at one wavelength, and then it emits light at another wavelength. So if you use something like ultraviolet, and what happens is these things glow. You've probably seen with, UV lamps, and you've got socks that glow, and this that glows, and that glows. What happens is the ultraviolet light is projected through the specimen, and the fluorescent molecules in the specimen emit the light to form an image. And these you've probably seen with larger eukaryotic cells where you have specific fluorescent molecules bind to actin or myosin, or they will attach to the nucleus. And so they give you a much greater understanding of cytoskeleton and other structures, okay? Then we have the confocal scanning laser microscope. There is a mirror, and it will scan a laser beam across successive regions of the specimen, and a computer reconstructs the image of the specimen. Now, we're going to move to electron microscopy. Electron microscopy was really incredible when it first came out. It was about 1935. And for a while beforehand, scientists understood they had a lot of evidence that suggested the existence of viruses, but they never saw one. And usually, you get that Missouri. I'm from Missouri. The show me stage. Show me, is it there? Well, they were. 1935, one of the groundbreaking developments with the onset of using electrons instead of light rays was that the electron beam wavelength is much smaller than the light waves. Therefore, your imaging of smaller structures can occur. The range of imaging is 1,000-fold better till about, you get down to about 0.3 nanometers. Now remember, nano is billionth. So what you're saying is even less than a billionth of a nanometer you can really see. How does this work? Well, the electron microscope, as you can see on this diagram here, really is sophisticated. And what does it do? You have electron beams going down toward the specimen, and you use magnets to help focus the beams. You will have an imaging film or screen that records the image. There are some disadvantages. You haven't seen an electron beam give a movie. Why? Well, the electron beam cannot work in air. You see, the electron beams would get interfered with the presence of air molecules. Therefore, when you set this up, the specimen has to be in a vacuum. A lot of times the specimen will have to be frozen or coated with heavy metal atoms to be viewed, because when the electron beams bounce off the heavy metal atoms, that's giving it the appearance of opaqueness. So usually what happens is, A, specimen's dead. So if you're looking for some video at the EM level, it's going to be very, very next to impossible unless you patch a bunch of pictures together. So it's very hard sometimes to view living processes in action. But what you can get are some incredible images, as you can see there. And you have several different formats of EM. The transmission electron microscope, which transmits a beam of electrons through the specimen. It allows for fine detail viewing of the cell interior. The scanning electron microscope. That's a beam of electrons that scans back and forth over the surface of the specimen. It allows for three-dimensional viewing of surfaces, such as what you see in that middle picture there. Okay? And then finally, the scanning tunneling one. This is allowing for views of structures at an atomic scale. You have a very sharp metallic probe that causes electrons to tunnel between the probe and the conductive surface. And so you're getting an incredibly fine detailed image, as you can see. First one on the top, that's transmission EM. One on the lower part, that's scanning electron. You can see scanning electron here, and right here you have a SARS virus, and you can get an incredible image of a scan via the scanning tunneling microscope. Now, next thing, microscope staining techniques. Now, I'm just giving you a heads up now. Take time to review this. It is either Lab 1, so that'll be week 1, or Lab 2, week 2 definitely. You're going to be doing some staining, so you need to get an understanding of some of the terminologies now. First off, what's a wet mount? It's a simple process. You take a drop of a liquid specimen, you place it on a microscopic slide, then cover it with a glass cover slip. Problem: the organisms might be transparent. How do you visualize nearly transparent cells? And this is important. You also want to be able to allow fast-moving cells to be visualized, to allow visualization of internal structures or internal cell biochemicals. You could be talking about a gas bubble. You could be talking about sulfur crystals inside. You could be talking about various inclusions. Or you could just be talking about structures like, endoplasmic reticulum, if you're talking about a eukaryotic. If you're talking about a simple prokaryotic like a bacteria, you probably want to see the nucleoid, which is the loop of DNA. So how do you get to do that? That's where we're going to get into the dye or stain as a specific application. Simple staining. Simple staining employs only one dye to that. The basic dyes usually carry a positive charge. Reason? You're going to go after and be attracted and stick to negatively charged molecules such as DNA or certain proteins. So the dyes you would be looking at are methylene blue, crystal violet, safranin, malachite green. Acidic dyes, these are used to stain backgrounds against which colorless cells would be seen. This is great because this is what they refer to as negative staining. You're staining the background, not the actual image of the structure. Now, the dyes are negatively charged, so they would be attracted to positively charged substances like histones, so you can get them inside in certain situations. So what are the steps? You take a drop of liquid with the cells, and it's added to the slide. Now, this film of cells in a solution is called a smear. The cells are fixed to the slide by gentle heating of the slide, usually by a flame. It's almost like you just pass the slide over the flame very briefly, like a couple seconds. Okay? You don't want to cook the bacteria. You want to just attach them. The slide then has the stain added. The dye is washed off with water. That's the excess. Some dye will get into the cell or onto the cell. You want to also note that there is an adverse effect that can happen sometimes. If you do too much heating, or the staining or the heating may distort the cell's shape. So you have to be aware of that possibility. Now we're going to get into what is called differential staining. This is where you're going to have two or more different types of stain. They're going to help distinguish one cell type from the other. The process takes advantage of the fact that different cells have different chemical makeups in their structure, and that may or may not hold different types of staining dyes. Two of the best known: gram stain and acid-fast. Well, let's get into this. First off, gram stain was developed by Dr. Hans Christian Gram, and I encourage you to go over that glimpse of history on page 45 and read it. You add a primary stain, it's usually crystal violet, to the smear. Now remember, we've already taken the smear, we've already heated it, we've already gotten it ready. You rinse the smear to remove the excess crystal violet. Now you're going to add a material called Gram's iodide. The iodide acts as a mordant. Now, a mordant is a substance that increases the affinity of the stain for cellular components of the dye. Okay? So it helps it stick there. The iodide and crystal violet form complexes. Therefore, it decreases the solubility of the dye, so it's fixing the dye to the stain structures. Now you rinse again. You're going to remove any of the excess iodide. When you're rinsing, you're going to add either alcohol or an alcohol-acetone mix. Now, these solvents are going to act as decolorizing agents. They're going to remove stain from the gram-negative cells. Okay? Now you're going to add a counter stain, because what happens if your cells are gram-negative? They weren't going to hold that stain, so how are you going to be able to still see them? So you use a counter stain, in this case, serafranin. Gram-positive cells appear purple. This is key point. Serafranin is not seen. Gram-negative, which lost crystal violet, appears pink from the serafranin. And here you see this all in the steps: crystal violet, the mordant, the decolorizer, and the serafranin, the counter stain. And you see in this image, you have gram-positive, those are the purple or bluish ones, and you have gram-negative, those are the kind of pink ones in the background. It is important that you keep this in mind. This is best done using fresh cultures. Now, what about acid-fast stain? Acid-fast is extremely important today. Why? If I say one word, tuberculosis, you'll understand. It has risen its ugly head, and it threatens many societies across the world. We use acid-fast to stain a small group of organisms that do not readily take up stains. These include members of the Mycobacterium. That includes Mycobacterium tuberculei, Mycobacterium leprae, basically those that cause tuberculosis and leprosy. Because these cells have a high lipid content in their cell membranes, to be honest, what they have are more waxy-like lipids, and it makes it really, really difficult. But it also accounts for their slow growth. But at the same time, you've got to have an answer, especially if you're trying to make a determination from some sputum, does this or does this not person have active tuberculosis? So, let's go through this. You take your sample. You have a high concentrated stain. The red dye is called carbol fusin. You have a brief rinsing, and then you flood with acid alcohol. This is the decolorizing agent. The cells that are acid-fast will still hold the stain. Then you use a counterstain of methylene blue. Now, acid-fast cells, they'll appear bright reddish pink. Okay? The non-acid-fast cells will appear blue. And you can see there are some cells in here. You've got some in here and some of the other little points that are pink. Okay, I had to take a brief break there for a second, but let's just continue. What we're looking here at is capsule stain. Now, one of the interesting things with the capsule stain is you're using some material that's really doing a negative stain. In other words, you're coloring the environment, the background, so that you can clearly see the structure that are sitting in the presence of the background. But the stain doesn't attach to the cells. It attaches to the background. This is called a negative stain. The reason being is that the cells have this capsule. It's a viscous layer outside of the cell wall. It's glycocalyx. One type that's used in these wet mounts is material called Indian ink, otherwise called nigrosin. And basically what it allows you to do is it darkens up the background so that you can see the capsule. If you notice, here's the cell, and there is the glycocalyx. And that's important because sometimes glycocalyxes present on certain cells will interfere with the capability of the immune system to destroy these cells. Okay? Here's another image of the same thing. Capsule stain here. You can see the black dark background. The sphere in the center is the actual cell. The outer part is this capsule. Moving on from there, we're going to go to endospore stains. As I mentioned, the endospores are going to be extremely important. You're going to go through this a lot through the course. But here's the thing. If you have a shell or a protective barrier that basically protects the cell, you have to find a way to adequately get the stain through into the dormant cell. The stain is used to identify dormant cells. It requires heat to facilitate the dye passing through the spore wall. So you're going to use as a primary stain malachite green, which gives kind of a greenish look. Then what you do is you take the smear and you heat it. Then you wash, rinse, so that's removing the excess, and then counterstain with serafranin. So non-endospore cells appear reddish or pink. Endospores you can clearly see there, sort of like a green in the center. These are all endospores here, 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 here. Now again, as I said to you before, cells that produce endospores include Bacillus, Clostridium. We're going to go through this with diseases and how they're important later. Now, what about Flagella stains? Wow. You can actually stain the flagella to be able to visualize it. The technique is difficult, requires mordants to help the stain adhere to the flagella. The arrangement of flagellas can be just like you'd think about like a sperm cell, you know, sort of attached at one end and just propelling the cell. But that doesn't always happen. You end up with flagellas being around the cell surface. They're called paratricheous, as opposed to polar, where the flagella is attached at the end. You can see over here, this is where the flagella is at the end of the cell. These are paratricheous, and the reason being is that they will cause the cells to wiggle all over the place in bizarre searches for nutrients, as opposed to going in one straight direction. The next image, which I kind of jumped ahead there for a second, sorry about that, are fluorescent dyes. Now, the neat thing about this is that you're going to be able to see the dyes in action where they attach to parts of the cell, and when they're exposed to certain light, this is going to basically give off a certain different color light. It's been used particularly to detect living versus non-living cells. And the dyes, as I said, mention select certain cell structures. So what you've got are these various unique types: CTC, acridine orange, calcifluor white. Calcifluor white is used for fungal cell walls. Auramine, rhodamine. Rhodamine has been used for mycobacterium. Another type is immunofluorescence. Now, what's happening there is you have antibodies that will attach to specific cells or to the cell components. The antibodies are going to have the fluorescent molecules attached to them, and they will light up when they're exposed to a particular light frequency. And that group right here have fluorescent antibodies attached, and so you can identify the particular cells that you're looking for. Remember that, and we'll go through this later, antibodies are extremely helpful for you to do quick and convenient identification of specific antigens or proteins or markers on the cell surface. So if you've got a mixed smear of bacteria, you can pull out the ones you're really looking for, if they are there at all. Now let's talk about morphology of prokaryotic cells. Morph basically refers to shape or cell. You'll see this on, of course, page 57 of your book. You got to keep in mind that morphology is really the classification of bacteria being done by shape. But lots of bacteria may have similar shapes, so we have to go further down in other means of identification. We'll get into that in a bit. But you need to know the terms. Cocci is sort of a rounded, and then you have rod, which is kind of a rod, rounded edged structure. Vibro is kind of looks like a comma. Spirillum is sort of a corkscrew. It's sort of more of a wave, because the spirochete is more the corkscrew type. Okay? Coccobacillus is an interesting combination where basically it's somewhat rounded, but then it is sort of rod-shaped at the same time. And here you see some of the other types. Again, here's the Coccobacillus, as you can see right over here, and it's kind of rounded here. Whereas with the rod shape, the bacillus, you can see it's more almost like a boxy type of appearance. One of the means to identify Bacillus anthracis is what they call boxcars, because they're going to be attached to each other. They're going to be rod shapes attached to each other. Vibro, you can see it looks like a comma or a, you might say a boomerang or a curled structure. Spirillum is just wavy, but the really tight curls are in the spirochetes. Okay, you need to know this. There's also pleomorphic, which means many shapes. The bacteria just don't have a set configuration. You'll see this. Sometimes a prothescate is a star-like cytoplasmic extensions from the bacteria cell, and you can see that there. But then we went from just shapes to groupings. Okay? And in the groupings, this is where you have to deal with issues of, well, how do they reproduce to do this? Well, what happens is this. You're going to have to start really thinking this over. Binary fission is a way in which the cells basically go from one and divide into two. Okay? There's not a lot of playing around that you would see with mitosis of exchanges. You don't have the PMAT, prophase, metaphase, anaphase, telophase. You just build up in a certain amount of volume inside, and the cells will divide. But what happens is when the cell divides, it doesn't necessarily dissociate into two separate cells. They'll stay connected. That's where you get Diplococcus, di, 2. Notice that on the lower part there of the image, you have the chain of cocci, okay? And then you sometimes will get packets. There'll be clusters, sometimes clusters of 8, sometimes clusters of 4, okay? Clustering will occur if you have cell divisions where they have several, what is called division planes, and they're very random. So it could be, you know, in the X plane, the Y plane, the Z plane, in between, etc. Okay? The packets, you're going to get it dividing in 2 or more planes perpendicular to one another. The clusters, you're going to have it all over the place, and you can see that. And the cluster, this is where you get into, you see that last image there down in the lower right. That's Staphylococcus aureus, the cluster of grapes, the golden grapes. Okay? Then we also have to deal with multicellular associations. Some cells are going to live in associations. The associations may lead to the development of unique structures. Mycobacteria will glide over surfaces and move in packs. So in other words, a single cell you won't have. You will have cells organized to ensure the survival of the species. During shortages of water and nutrients, they will form this mass, forming a fruiting body where the interior cells enter a dormant state until favorable conditions for growth exist. This is where you're also going to get into the forming of biofilms, cluster associations where bacteria cells are basically encased in a polysaccharide coat. Biofilms are a big no-no in the medical field, particularly with catheters and implants, etc. Because where the bacteria are, that basic biofilm around them protects them from the adverse effects of antibiotics. Sometimes it requires the removal of the catheter or the actual implant to stop the infection that's going on. Now, you can see here the basic structure of a bacteria cell, a prokaryotic cell. Keep in mind a couple of things. There's no nuclear membrane. There's no membrane-bound organelles. You will find ribosomes, essential for protein synthesis. You will find that the cytoplasmic membrane, cell wall, and a capsule. Sometimes they're put together to make up the entire cell envelope. You will have a cytoplasm. Think of it as a big stew, in the sense that floating around in there are dissolved gases, water, sugars, wastes, proteins, enzymes, etc. No sequestering of any of this stuff. You will have the nucleoid, which is sort of the coiled up form of the DNA. You may also find, besides the ribosomes, you'll find pili or flagella. You may actually find storage granules that may contain things like sulfur or phosphates, or they may have some type of a lipid. Okay? Let's move forward. As we talk about the cytoplasmic membrane and the cell structures of the prokaryote, these are things that you're going to need to know. I encourage you to spend time on table 3.3. Keep in mind that when we first looked at cells, if you remember in A and P, you had, well, cell membrane, okay? The cytoplasmic membrane. Final exchange site between the cytoplasm and the cell exterior. Yeah, but something a little bit more complex is also there. Do keep in mind that in the cell membrane, you're going to have a phospholipid bilayer. You're going to have some proteins that pass all the way through that bilayer, some that do not. The phospholipid bilayer has basically a hydrophilic head and a hydrophobic tail. Okay? But, yes, there's more. What does that mean? That affects a lot of things. Certain things will be able to flow through, certain things will not be able to flow through. And also those proteins may act as transporters, they may act as cell receptors. And so we have to also look at something else here, the difference between bacteria and archaea. If you look at bacteria back here, the bacteria phospholipids have fatty acid tails. Archaea have different side chain linkages to glycerol and do not have fatty acids. They have attachments to hydrocarbons. And what I mean by that are just long chains of hydrocarbons. There's no oxygen, and it's a very different arrangement. What is the basic purpose of that cell membrane to control molecular exchange? Simple diffusion, osmosis may occur, but how does one resist osmotic pressure to the bacteria cell? That's where we get into the cell wall next. Remember that water can pass through a membrane transporter called aquaporins. Remember that nonpolar small molecules such as gases like CO2, O2, nitrogen, and very small hydrophobic molecules can pass through. What cannot pass through are large molecules, sugar, ion, amino acids, ATP, macromolecules, etc. And so we start getting into these challenges of transport. Okay? We're going to get into the cell membrane in a minute, the cell wall, I should say. But another interesting thing about the cell membrane, Besides controlling water flow in and out, because remember, you're going to be exposed to hypotonic solutions on the outside or hypertonic solutions on the inside, you have to control all of this. And this control system requires energy. The cell membrane also has an electron transport chain. The electron transport complexes pump proteins, that's hydrogen ions, out of the cell to create an electrochemical gradient. Now, if you remember dealing with this for the mitochondria, you will probably be ahead of things. What you have to keep in mind is this: the gradient will drive cellular processes like transport mechanisms, flagella movement, and ATP synthesis. As the hydrogens start leaking back through or they're being pumped out, what is going to create this gradient is going to be the energy source to create molecules like ATP, etc. Now, we have to deal with types of transport mechanisms. We're going to have facilitated diffusion, which you probably are familiar with if you remember your AP1. Although it's rarely used in prokaryotes, basically facilitated diffusion takes advantage of a concentration gradient. So you have one side is high concentration, one side is low. But what you have to have is either a channel or a gate molecule that will allow the molecules to come in and, in some cases, go out wherever possible. But then what happens if you're going against the concentration gradient? In other words, what you want to do is you want to bring in molecules that there's a low concentration on the outside, there's a high one on the inside. That's where you're going to have pumps or transporters, and those require energy. Part of it is going to be taken not just by using ATP, but by the protons that you saw back here. Okay? And the ABC transporters are going to be using ATP. They will bind, their binding proteins deliver a molecule to the transporter. So, what do we deal with here? Okay, take a look here. Given transport molecule recognizes the specific molecule, molecule goes in. Okay, no problem. Now, facilitated diffusion, you have a high concentration out here, low concentration here, but when this integral protein in the membrane allows the particles to go through, and it opens up, fine. You have transport from a high concentration to a low. Now, active transport using a proton motive force, what's going to happen is you get a low concentration of particles that you want inside. There's a high concentration here. What's going to happen is they're going to piggyback together the excess amount of hydrogens here and push through. You have to have a pump to go from low concentration to high concentration for transport of molecules across a barrier. Just like if you had flooding in your basement, what do? You call out the pump. And what does the pump do? It pumps water from a lower area of your basement outside. To do that requires electrical energy. Same thing here. In this second type of active transport, you are going to have a binding protein if necessary, take the molecules, run them through, but the process of going through this pump requires ATP, and it's going to go ATP, it's going to get hydrolyzed, you're going to lose one of the phosphates, and you're going to end up with ADP. What about group translocation? The transporter chemically alters the substance as it is transported across the membrane. So when this molecule comes in here, it gets phosphorylated, if you see there where the P is. Therefore, it's not in the same group here. It's not a high concentration here. It's still actually higher out here than here. This happens also in eukaryotic systems. When we have muscle cells, they bring in the glucose, they immediately begin phosphorylating it to keep it inside. Okay? Finally, we have also secretion, and secretion is a process used to secrete proteins. So we're talking about going outside now. Okay? Now, in that case, secretion requires a signal sequence on the protein of about 20 hydrophobic amino acids, and sometimes that is used to used for the release of the extracellular enzymes. Okay? And we've gone through this here. Okay. Now, and by the way, if at any time, remember that we have a chat each week, so if you have any questions, you're a little fuzzy on something, we can discuss it then. Again, review the microassessment on these principles. Let's go to a cell wall. Now, this is where we come back to gram-positive, gram-negative. If you walk away from microbiology with one thing, you'll be able to understand what gram-positive, gram-negative means, what bacteria have them, and why do they appear as gram-positive or gram-negative. That's why I want you to review this, okay? So if you take a look at 3.5, you got a comparison there. Keep in mind that antimicrobial medications target some of these structures as a means to kill the infection. Keep in mind that we talked about this. The cell wall helps the bacteria withstand forces that occur when you have hypertonic or hypotonic concentrations outside of the membrane. If you have such disparities, you don't want the cell to lose all its water. You don't want the response to be that it sucks up so much water that it bursts. If it would inflate, it would be held back from completely inflating and then bursting by the cell wall. But what's the walls made up of? When you're gram-positive, you have a thick peptidoglycan layer. Teichoic acids are present, and you have no periplasm. With the gram-negative, you have a thin peptidoglycan layer, no teichoic acids, and the periplasm is present. Here is the image that you're looking at for the wall. And this image, first off, is the components and structure of the peptidoglycan. Now, notice the arrangement. Notice the chains. Notice these unique molecules here. And they're identified, okay? This is a macromolecule composed of alternating series of N-acetyl muranic acid and N-acetyl glucosamine. So NAMs and NAGs, and they're attached to NAM. There's going to be a tetrapeptide chain of unique amino acids. Okay, so it's a big mesh. And for gram-positive cells, the tetrapeptide chains are attached by a peptide interbridge, which the sequence varies according to the different species of bacteria. So let me help you to understand a little bit better here. Take a second here. Gram-positive cell walls, for example, Bacillus, Staphylococcus, Streptococcus. The cell wall is going to hold a gram stain due to a thick layer of peptidoglycan and teichoic acids. Yes, you can laugh at it. It's okay. Everybody stumbles on their tongue. Teichoic acids include chains of ribitol phosphate and glycerol phosphate. Some have lipid attached, so they're liptoteichoic acids. These acids stick out of the cell wall. They're negatively charged and give the gram-positive cells their negative charge. And you can see this all the way through here, as well as through these structures. Okay? Now, what about gram-negative cell wall? These include Escherichia, Neisseria, Pseudomonas. The cell wall contains a periplasm. Okay, and you can see that labeled there where the periplasm is right over here. The thin peptidoglycan layer, and it has an outer membrane that requires porin molecules for molecular passage. Okay, here's the porin molecule here. Keep that in mind because sometimes certain porins can also play roles as drug pumps, thereby making the antibiotic not as effective in killing the bacteria because it's being pumped out of the cell. So, the periplasm is the region between the cytoplasm membrane and the outer membrane, and it's filled with a gel-like fluid called periplasm. ABC transporters and peptide-degrading enzymes are located in the periplasm. Okay? Outer membrane is made up of a lipid bilayer containing, except the lipopolysaccharides, the LPS, are present, not phospholipids. Okay? This is your lipopolysaccharide here. This is important too because when the cells are breaking apart, this is where you get into what is referred to as endotoxin products. The body reacts negatively to LPS. So, it will elicit symptoms similar to the bacteria infection in animals. There is a particular syndrome called Jauregg-Herxheimer reaction. This is a reaction to endotoxin-like products released by the death of harmful microorganisms within the body during antibiotic treatment. In other words, you start putting them on the antibiotic, they seem to be getting worse. Doc, what's going on? This is the Jauregg-Herxheimer reaction. The good news is that it's indicating that the antibiotic is killing the microbes. The bad news is they're going to have to ride out the storm with these LPSs flowing and causing and reacting to the LPSs. Okay? And this may result in a systemic inflammatory response. Usually it's not life-threatening, but if it starts appearing, you want to monitor the patient a little bit more closely. This was classically associated with penicillin treatments of syphilis. Now, porins are specialized proteins. They allow small molecules and ions to enter in and out of the periplasm. The LPS is made up of several molecules. You can see this listed up here. It is lipid A, and that's a lipid embedded in the lipid bilayer, O-specific polysaccharide side chain, and the component, the sugar chain component that is used to identify select strains of bacteria. Are we pretty clear? And you can see here is an EM of that entire setup: outer membrane, cytoplasmic membrane, periplasm, and peptidoglycan. No problem there. Here is an up-close image of the LPS. Now we're going to talk for a second about when you go through the staining and everything else that's going on, what happens. The differences are very important. And by the way, antibacterial compounds, well, this gets you into the structure. Penicillin binds to proteins involved in the cell wall synthesis. It prevents cross-linking of adjacent glycan chains, and that's how penicillin basically works. Lysozyme, this is an enzyme that breaks the bonds that link NAG and NAM molecules, and thus loosens the structural integrity of the glycan chains. Lysozyme is something that you'd find in tears. You'll also find it in abundance in eggs, etc. Okay? The difference in the staining. The peptidoglycan layer plays a major role. With the alcohol treatment, the thick peptidoglycan layer dehydrates and retains the dye. The thin layer does not. It can't hold it. And therefore, the first stain leaves. And you can see this in the gram-neg versus gram-positive after decolorization. Okay? You've got dehydrated peptidoglycan, the damaged cytoplasmic membrane, but the crystal violet iodide complexes will not be easily washed out of the cell, whereas in the thin peptidoglycan, they will be. And that's when we move into using sarafranin as the secondary stain. Okay, but what about bacteria that lack a cell wall? Here's an example, okay? And that's not easy when you really look at that. You're probably sitting going, what is that? Well, that's Mycoplasma pneumoniae. They're flexible, they lack a cell wall. You might call them pleomorphic. They don't really have a set shape. So, the cells have a stronger cell membrane, since their cell membrane contains sterols. Okay? Examples of sterols include cholesterol, eggrosterol. Eggrosterol you'll find in fungi. The sterols, because of their planar structure, tend to make the membrane stiffer, less pliant. If you remember AP1, for those of you that had me in AP1, I talked about that. In eukaryotic cells, 75% of the lipids are phospholipids. It's the 25% that you can have making the cell walls, cell membranes, much more inflexible, much more stiffer by having higher amounts of cholesterol present. Okay? Now we've talked about that, but what about archaea cell walls? They have a variety of cell walls. Some exhibit a similar molecule called pseudopeptidoglycan. Some have S layers, that's sheets of flat proteins and glycoprotein subunits. That's where I would encourage you to take time to review the microassessment. Now, structures outside the cell wall. What are we talking about there? Well, bacteria have a protective or attachment gel-like layer called the glycocalyx. We've talked about this. This is the capsule. If the glycocalyx is distinct and gelatinous, we might call it a slime layer if it's more diffuse and irregular. The composition varies among species. And this is where I was talking about earlier, biofilms. And it's important in the determination of whether the strain is infectious or not. The capsule can protect bacteria from the body's defenses. In teeth, we have biofilm, and that's caused by Streptococcus mutans. In bacterial pneumonia, the capsule for Streptococcus pneumoniae protects against host immune defenses, as I mentioned that before. The next we want to talk about are filamentous protein appendages. Now, bacteria have structures that protrude from the surface. Yeah, we know that. It may have one polar, or it may have flagella distributed over the entire surface. That's where we get to paratrichius. The top one you can see is clearly paratrichius. The lower one seems to be just polar. This is all powered by proton motive force. Basal body anchors the flagella to the cell wall and the cytoplasmic membrane. The hook is the connecting filament to the cell surface, and the filament is composed of flagellin. You see another case of this here. That's paratrichius on the left and polar on the right. Okay? These are all used for locomotion, and they can direct the organism towards or away substances. This is part of chemotaxis. Organism runs with a directed motion and spins in a tumble or self-directed or to self-direct or reorient itself. Okay, here's the hook, the filament. Here are all the flagellum. This is paratrichius. Okay? Here's a tumble. They all spin, and then they all get coordinated, and then they tumble. Now you might say, what the heck is tumbling got anything to do with it? A lot. Let's say you're moving toward a gradient, a buildup of certain nutrients, amino acids, lipids, or you're moving away from something, a concentration of antibiotic or some hostile chemical or a change in pH. What you have is sort of a run for a distance, tumble, spin, run for a distance, tumble, spin, and this continues. Every time you make a major turn. Now, I got a little idea to share with you. If any of you have a Roomba or have seen Roombas in action, they do a similar process. Basically, when they come and they bump something, they go and do a spin to make sure that they clear it, almost like a tumble approach. And then they go for a distance straight. And if they bump into something, they do a tumble and a spin. And it's the same approach. The idea is to try to maximize your space coverage in a three-dimensional array so that you can maximize your orientation toward the best opportunities of nutrients or away from hostile or dangerous things. There are other types of taxis, by the way. Orientation toward light, phototaxis. Orientation toward levels of oxygen, aerotaxis. Magnetotaxis is taxis toward magnetic fields. There are some bacteria that actually have small magnetite particles linked together inside their cytoplasm, and it acts as an internal navigation system. Here is the perfect example. As you can see, those dark dots are magnetite particles, and they help orient that particular organism to its magnetic fields, and thereby know where to go. Pili are thinner, shorter filaments. Their function is to enable cells to attach to surfaces, and they're called fimbriae. We'll get into this later on when we talk about how bacteria mate or connect with each other and exchange small amounts of DNA. Okay, that I believe is going to be in chapters 6 and 7, if I'm not mistaken. But along the tip of the pili is a molecule for adhesion. And this is an attachment molecule. It's important in some pathogens for attaching and remaining in the small intestine. In other words, how to prevent the cells from being sloughed off during a massive, purge that might occur. Basically, it would just attach to the surface of the cells with this pili. Now, it's also useful in conjugation exchange of DNA, and I mentioned this before. That's why they call some of these pili sex pili. And they're also useful in locomotion in solid media. They have a twitching motility and a gliding motility that allows them to pass through, such as going through some types of gels like agar gel, etc. Okay, now where are we? Well, we're going to deal with the internal structures of bacteria. I do encourage you again to review the microassessment. You got to keep in mind that prokaryotes have some structures internally, but mostly the enzymes and other substances are mixed in that cytoplasm. That's why I call it a stew. It's literally lots of stuff all mixed together. And so because of the small space, they have a high probability of bumping into the right items. But you still have to have somewhere your chromosome of DNA. And that gel-like region of supercoiled DNA is called the nucleoid. And you can see in the dark area on the red, on the image on the left. And you can see how they split open the cell, and you see the spilling out of this uncoiling occurring on the right there. Okay? Now, the next issue is, besides this supercoiled DNA called the nucleoid, we're going to deal with, you thought you were just going to deal with ribosomes. Well, first you've got to keep in mind about something else. And this is critical. We're going to get into this much more later on in Chapter 8. There are these smaller rings of DNA that are not attached to the chromosome. They convey various genetic traits. They may include drug resistance, endotoxins, exotoxins, etc. A single cell may carry hundreds of plasmids, and they can carry several hundred genes on each plasmid. This is something we're going to come back to a bunch of different times. Plasmids are absolutely important that you need to get familiarized with. Aside of plasmids, we now come into the part of how we make proteins. And that, again, just like in eukaryotes, are ribosomes, but they are a little different in size. They are a little bit different in composition. But they are the protein synthesis sites, and they are composed of two subunits, a 30S and a 50S. When they combine, they're referred to as a 70S. Now, the next area are storage granules. And I've mentioned this before. Storage granules are really important. They contain accumulations of high molecular weight polymers, starch, glycogen, poly-B hydroxybutyrate. That's a mouthful, but it's important. Volutin, which is a form of phosphate storage, and you can identify it by staining the cells, and they will appear red on the exposure of methylene blue. That's why they're called metachromatic granules. Also, you'll find fat bodies as some types of storage granules. Besides storage granules, what about gas? Yes, gas. Small, rigid protein structures hold gas and provide buoyancy for aquatic bacteria. You can see those gas vesicles right there. And then we get into endospores. Now, endospores is the dormant cell type, and it's produced by a sporulation process. Spores are resistant to UV light, heat, desiccation, meaning drying out, toxic chemicals, etc. They can germinate to form a living active cell, the vegetative cell. So when we talk about spore, it's a dormant cell type. The active living cell is the vegetative cell. You will find these in Bacillus and Clostridium genera bacteria. To see the process, I would encourage you to go to 346. Here's another image of an endospore. Here's the process. What happens is this. The vegetative growth stops. DNA is duplicated. A septum, a wall forms there, and you can see that. And the cell divides asymmetrically. One part gets a lot of the cytoplasm, etc., but the other, the smaller cell is called the forespore. The larger cell, the mother cell, engulfs around the smaller cell. The forespore, that smaller one, has a layer of peptidoglycan laid between two membranes. The other cell is making proteins that form the spore coat. The mother cell degrades, dies out, and the endospore is released. Now, this is extremely important. We're going to be going through various types of diseases, and people would say, well, I cooked it and I still got sick. Well, that's because the Clostridium that might have been in, the Clostridium botulinum, for example, can be in spores, which is why you have to go to higher heat and higher pressure to kill them in the canning process. And if you don't, you've got Clostridium spores that are consumed, that are viable, and that when they enter the gastrointestinal system, they are reactivated and start issuing out live Clostridium botulism bacteria, which will, of course, release botulism toxin, and then you have a dead patient. Review the microassessment. Now let's go to eukaryotic cells. In a summary, you see this listing, and these images are somewhat familiar, at least the top one should be, for most students who've had A and P. What happens with a eukaryotic cell? The big changes are membrane-bound structures. You've got a membrane-bound nucleus. The DNA is only in the nucleus. The DNA always stays in the nucleus. Copies of the genes are made in RNA, and they pass outside the nucleus where they are formed by ribosomes. Some of the ribosomes are attached to a membrane-bound structure known as the rough endoplasmic reticulum. We can continue on that. Organelles, the little organs. These are many membrane-bound structures. The nucleus is also membrane-bound, as I mentioned. Basically, what you have is a modified system to transport and store some materials. Some of them will be in vesicles, membrane-bound sacs. I encourage you to take the time to review all of the membrane-bound organelles. You'd see this on table 3.6. Of the functions of these organelles, as well as the internal protein structures, the ones that are not bound by membrane-bound organelles, because you really need to know this. The greater the difference between eukaryotic and prokaryotic means the greater the chances you can develop a treatment for a prokaryotic disease, and it would not interfere with the biochemistry or physiology of the eukaryotic organism. Remember that. The plasma membrane. The lipid bilayer is 75% glycoprotein, some cholesterol, other lipids. Fungal cells will use ergosterol. You will find in that plasma membrane receptors, mostly glycoproteins. It's involved with cell signaling that may require secondary messengers. Cells from plants and fungi have cell walls, such as you see here. Here's a plant. The plant cell membrane is on the inside, the cell wall is on the outside. Cell wall is consisting of, for plants, lignins and cellulose. Transportation processes. Well, you still have to deal with diffusion, osmosis, and facilitated diffusion, which is via gate, channel, and carrier proteins on the plasma membrane. Active transport is via pumps that use ion gradients or use ATP to pump substances through pump proteins. You also have the amazing processes such as endocytosis, and that would include phagocytosis, where the cell can use arm-like cytoplasmic extensions, and encapsulate, surround, and enclose the item and bring it inside. Pinocytosis, such as what you see in these, and that's bringing in liquid material. And receptor-mediated endocytosis, which you see on the far left there. The substances are bound to the receptors, and then you have a pinching in, a cratering in of all of these receptors with bound target material, which form an endosome, go inside, and are broken down. And the substances are released and used in the cell. You also have the process of exocytosis, which is by secretion. When you look at structures such as cytoskeleton, you have microtubules, microfilaments. Many microfilaments are basically actin filaments made of actin subunits, and intermediate fibers. These filaments are also essential. This is what helps provide, it's crazy to say, but people had a hard time believing it, a cellular skeletal framework. Then you have structures on the outside. And those structures include flagella. Usually for eukaryotic cells, you're going to have 1 to 2 per cell. They function in locomotion, the transport of the cell. Cilia, many are on the cell, and they're shorter than the flagella. Those are usually used to transport substances past the cell. Or, for example, with protozoa, you'll see them all over along the wall, the cell membrane, and they help propel the entire cell, the protozoa, because it's a one-celled organism, through the liquid medium. Now you notice that you've got sort of a skeletal structure inside the flagellum. That's going to be microtubules. This is going to kind of coincide with the next area that you have to review, which is Perspective 3.1. Now, 3.1 brings up an interesting thing. Pathogens have the capability of hijacking actin. This is the first situation you have. It's called ruffling. Salmonella will enter the intestinal epithelial cells. How they do this is literally they flatten out the microvilli in the entire surface, and they make it sort of like a landing pad. This causes ruffles that form, that eventually enclose the bacteria cells. It allows the means for the cells to get inside. The second situation is actin-based motility. Once Listeria cells are inside of the host cells, they will begin to polymerize at one end of the bacteria cell actin. This creates sort of a propulsion vehicle, a chain or a train track that will help push them along through the cell, and with enough force that it can actually propel itself through one cell into the neighboring cell. And that's critical. The last one is pedestal formation. Certain E. coli cells will actually alter the intestinal epithelial cells, alter that cytoplasmic membrane, create sort of this pad or pedestal. And this is causing also the underlying structure of the host cells, actin, to help rearrange and establish this pedestal where the bacteria cell, E. coli, will attach to. Okay? Let's finish up with some of the other structures of a eukaryotic cell. Of course, the nucleus, the site for the storage of DNA. Now, DNA is going to be wrapped up by histone proteins and controlled and supercoiled into chromosomes. How does that occur? This blue structures are histone proteins. They kind of form sort of the spool, and DNA will spiral around it once, twice, and then go off. And so what you have here between the histone and the DNA is a structure called a nucleosome. These will all begin coiling up and then coiling up further until you form the chromosomes that you are very familiar with seeing. Oh yes, and by the way, how do we get substances in and out of this nuclear membrane? You have pores here. I want you to keep that in mind, because the pores will allow products to go in and out. For example, messenger RNA to go out from the inside of the cell, ribosomes and certain other smaller pieces of messenger of transport RNA and ribosomal RNA, etc., to get transported out, but DNA stays inside. Now, from there, we have to get to our energy powerhouses, the energy production organelles for the cells. Mitochondria. You see mitochondria both in plants and in animals. Animals don't have chloroplasts. Mitochondria produce ATP via citric acid cycle and the electron transport chain. If you're not familiar with that, you might have heard of it as the Krebs cycle. And what you have to keep in mind is all of this is occurring on the inside through the inner membrane. Interestingly enough, the mitochondria have rings of DNA that code only for mitochondrial proteins. And so this gets us into the endosymbiotic theory, where basically they figure that a larger cell engulfed a smaller cell, and instead of consuming it, they developed a symbiotic, a living together arrangement. I'll give you energy, you give me shelter and nutrients, and everybody's happy. And that seems to work out. The chloroplasts are believed to have done something in a similar way. What chloroplasts are, they use energy from the sun to convert carbon dioxide and water to glucose and oxygen. The cells then use the glucose for food energy. And interestingly enough, this is what you see. You see the discs, these thylakoid discs. They have an inner and an outer membrane, but in those thylakoid discs and inside of those structures, there are enzymes and there are pigments that grab onto the light energy and transfer this energy into certain biochemical processes. This is in a full photosynthesis biology lab, but just to give you sufficient information that by doing these processes, you eventually construct from carbon dioxide and water glucose, and you also will release oxygen. Okay? Now, let's not forget some of the other membrane-bound structures as we finish up here. The endoplasmic reticulum. You have the rough endoplasmic reticulum in eukaryotes that processes and packages proteins for subsequent export out of the cell. They are rough because they're studded with lots of ribosomes that inject the developing protein into the lumen of the RER, and then it's processed further. Now, it's smooth. These organelles are different. They detoxify certain chemicals and do packaging and synthesis of lipids. Okay? The Golgi apparatus, or the Golgi process as it's been called, is a system which packages proteins for final export out of the cell. So once you form this modified protein in the rough endoplasmic reticulum, it will be transported out by a little vesicle, a membrane-bound packet, and that will eventually knead up, fuse with the Golgi apparatus to be finally packaged properly. Any further chemical modifications that occur, and then once it bubbles off, forming a vesicle again, that vesicle will usually attach by exocytosis and release its content outside of the cell. There are one or two others that are very important to bring up right now. Lysosomes and peroxisomes. Lysosomes are organelles that contain very strong enzymes and are used to digest, break down old organelles and food trapped in vesicles. Now that would include what you see in white blood cells when they engulf a bacteria and bring it inside. They'll have it in a packet. The lysosome will fuse with that packet, and the digestive enzymes will destroy the bacteria inside under the best conditions. We'll get into host microbe interactions later on in the course. Peroxisomes contain enzymes that generate oxygen and superoxide molecules for breaking down lipids, detoxifying certain chemicals. Now, I encourage you to take time to review Table 3.7, the review for prokaryotes and eukaryotes. I also encourage you to go over the microassessment and always review the chapter summary. The chapter summary is a thumbnail sketch. It'll give you a good idea of what you have to face in the up and coming weeks or with that chapter. And with that, we are done with this particular lecture.